Now let's discuss about the famous Watson and Crick's model for DNA double helix. Watson and Crick produced the famous model in 1953. This work, the Nobel Prize given in 1962 to Watson, Crick and Wilkins in 1960. But before the Nobel Prize was awarded, Wilkins died of cancer at the age of 38 years. Which I had already discussed in the historic background. My previous lecture I have already mentioned. Now, what was the proposed 3D model? Point number 1, Watson and Crick proposed that DNA molecules consists of two polynucleotide chains. Two polynucleotide chains. Two polynucleotide chains. How single polynucleotide chain is formed in case of DNA, I had already described here. Two polynucleotide chains wound around each other in a right-handed double helix fashion. Right-handed double helix fashion. Right-handed means clockwise. Clockwise. And the two strands are anti-parallel. Two strands are anti-parallel, means opposite polarity. Opposite polarity. Opposite polarity means one strand will be moved from 5' to 3' and another 3' to 5'. One strand will be 5' to 3' and another will be, this one is known as opposite polarity. Is anti-parallel means anti-parallel. This one is known as opposite polarity, and what is anti-parallel means? Anti-parallel means head of one chain is against the tail of another chain. This one will be head and this one will be tail. The head of one chain here, head of one chain here, and tail of another chain is here. And this is known as anti-parallel and opposite polarity. The sugar phosphate backbone, the sugar phosphate backbone, the sugar phosphate are on the outer side of the double helix, are on the outer side of double helix. Point number, the bases oriented towards the central axis. The bases of both chains are flat structures oriented perpendicularly to the long axis of the DNA. This is the two polynucleotide chains together with hydrogen bonds. Bases are bonded with hydrogen bonds. This hydrogen bond, as all of you know, this is a weak chemical bond, not so strong. Weak chemical bond. A make double bond. These two are hydrogen bonds. Adenine make bond with T, and G, guanine, make bond, triple bond here with C. As all of you know, A for adenine, T for thymine, G for guanine, and C for cytosine. Here For the presence of triple H bond, melting point is high. For the GC percentage determine melting point. Okay. For this 3H bond, the melting point is high. So on the whole, how we can conclude it? That is purines pairs with pyrimidines. Weak chemical bond, hydrogen bond, it is very easy to separate the two strands. For example, by heating, by give heat, we can easily separate the two strands of DNA as there is weak H bond is present between these two strands. AT and GC base pair, only one can fit the physical dimensions of the helical model, and their arrangement according to Chargaff's rule. The specific AT and GC base pairs are called complementary base pairs. These are called complementary base pair. So, the nucleotide sequences of one strand can dictate the another strand nucleotide sequence. For example, if one strand have this sequence 5 prime TATT CCGA 3 prime, then the complementary strand will be 3 prime AAGGCT 5 prime. The base pairs are 0.34 nm apart in the DNA helix. Base pairs are 0.34 nm apart in the DNA helix. A complete turn, a 360-degree turn, which is known as complete turn of the helix takes 3.4 nanometer. Therefore, there are 10 base pairs per turn. And the point number 7, the external diameter of the helix is 2 nanometer. So, here I had already discussed the 7 points. Carefully look about the second point. I am going to erase this. Now, let's draw, after discussing the very important 7 points, now let's draw the famous Watson and Crick model of DNA double helix. So this is the famous Watson and Crick model. These two are backbones. These two are known as backbones. These one are base pairs. These one minor groove. This one is known as major groove, and here 0.34 nanometer from here to here, the 3.4 nanometer. If we draw a imaginary axis here, this one is known as axis of helix, and this axis of helix, it's not real, it's completely imaginary. And this is stylized diagram of DNA double helix. Previously I have already discussed about 7 major points. Now come to another points. Point number 8. There are 2 grooves are present, major groove and minor grooves. There are 2 grooves are present, major groove and minor groove. The minor groove is shallow and major groove will be deep. Point number 9. The length of one helical turn. Length of one helical turn or length of one pitch turn. One pitch turn is 34° angstrom, 3.4 nanometer. And what do you mean by this helical turn? Helical turn means, one pitch turn means, distance between 2 minor group. Here it is minor group, distance between 2 minor group. Distance between 2 minor groups. Number of base pairs. Number of base pairs in each helical turn is 10 base pair. 10 means 1, 2, 3, 4, 5 inward and 1, 2, 3, 4, 5. Total 10 base pair. Base means here, pair means 2. Okay. Number of base pairs in each helical turn is 10 BP. This one is 1 helical turn, here 1 pitch turn. Next, distance between two corresponding base pairs, that's equal pitch turn or 1 helical turn divided by number of base pairs in one helical turn. So 34 divided by 10, 3.4 angstrom, 0.34 nanometer. Is it clear? Then come to the next and last point. Point number 13. Diameter of DNA is 20 degree angstrom or 2 nanometer. What is the diameter of DNA? Distance between two phosphates. Let's draw phosphate, sugar, this is diameter of DNA 20 degree, and the length between these two sugars, this one is 11.1 degree angstrom. So it's all about Watson, the famous Watson and Crick's model and their descriptions. Keep it very well in your mind and set at your brain, as it is very much important in your syllabus and for examinations also.